Hello everyone, we are team number 5 and this is our STEM project, Napcare. The main problem we identify is the contamination or pollution of water ecosystems and as a consequence the endangerment of its species. Every year approximately 22 million tons of plastic and paper waste is generated and end up in oceans, rivers and lakes. Paper napkins represent a significant percentage of this waste. So the solution we found to address this problematic is creating a napkin prototype with the capacity of being biodegradable, eatable, functional, and mainly that when ingested protects fish against other toxic substances or material that contaminates water. Uh, the STEM project objective is to create and present a real prototype that is designed for easily degradation in water, uh, serve as safely food or aliment for fishes, and that once ingested helps trap and evacuate toxic substances also consumed by the fish. The materials and methods we will use is as a base, recycled cellulose reinforced with vegetable starch. This is for the napkin to be resistant and functional for real use, but at the same time easily degradable in water. Chitosan from the shells of the shrimps alginates and zeolite. This way, when fish ingested, does not be harmful for his organism.
Our contribution as a team is to clean water ecosystems and reduce the negative impact of human waste on aquatic life. These are the CTGs involved. The number 6 is clean water and sanitation, and the number 11 is sustainable cities and communities. How is chemistry related? Applying chemical principles and knowledge, we will analyze and determine the materials and compounds in order to make the napkin easily and biodegradable, but functionally and resistant.
In the presence of solid waste in aquatic ecosystems contributes to pollution, exposure of toxic substances to fishes and other types of aquatic species. Our project aims to transform a type of napkin into an alternative that, due its components, cellulose, starch, ketosine, alginate, cellulite, is biodegradable in water and edible for fish. Once ingested, it will capture and eliminate toxic and harmful substances. Key concepts: biodegradability, capable of material to be broken down by living organisms. Absorption, process by which molecules such as toxins or pathogens adhere to the surface of a solid or liquid, influenced by molecular forces and surface properties. Chelation, chemical process where a substance, a chelating agent, binds to a metal ion, forming a stable ring structure known as a chelate ring. pH-activated release, design principle where compounds remain stable in neutral conditions, but become active in acidic environments such as the digestive system. Relevant theories: absorption isotherms. This theory describes the maximum capacity and efficiency of heavy metals absorption by biopolymers. Design of edible biodegradable films show that films made from alginate and ketosine can provide strength while providing functional bioactive agents. Fish digestive chemistry explains how the acidic gastric pH of fish activates certain compounds enabling detoxification processes with the intestine.
Key literal support. It is divided by five mainly important points. One of them will be alginate-based materials have proven effective in absorbing heavy metals from water. Another one will be kerosene and its derivatives are widely studied for binding metals such as Pb, Cd, Cu, and He. Composite alginate kerosene beads with EDTA and enhance removal of toxic metals. Also, development of bioactive ribofilms combining biodegradability and active protections has been reported. Last studies on fish gastric pH confirm conditions that will be triggered the activation of protecting agent. Connection to risk. Connection to regime question. We maintain three mainly points again. One of them will be this scientific evidence demonstrate the feasibility of creating a biodegradable napkin that not only breaks down in water but also acts as a functional for fish. The compounds present in the napkin will be absorb toxic metals, which is the entire digestive tract of the fishes. Our prototype will be a solid solution to water contamination and protection of water-related ecosystems. How our DI differs from current solutions? Most exciting existing approaches focus on external water filters, plants, or biodegradable products with active functionality. Our project proposed a biodegradable napkin that is designed to behave actively with the fish organism by capturing the toxic metal during the digestion, or mainly by this process, which this concept is not currently used in environment solutions. This will be our theoretical framework bibliography, which is we maintain five important references. Chemical substances involved. Environmental problem are toxic substances presented in contaminated water. We maintain two mainly points. One, heavy metals. It constitutes with lead, mercury, cadmium, and arsenic. By the other hand, are the industrial pollutants that it contains plastic, paper, and cardboard. On the other hand, solution proposal that is our napkin prototype. It is constituted by biopolymers that it constitutes with alginate with his corresponding equation, and also kerosene again with his equation. And at last, the functional agents are cellulose with his equation and starch.
This is our prototype model with a height of 15 centimeters and a base of 15 centimeters and a thickness of 0.05 centimeters. This is a chart that demonstrates the degradation time in water. The following graph represents the amount of napkin mass percentage remaining in the water per hour transcoded. The prototype variable simulation. The napkin degradation time, the time is required from the biodegradable napkin to break down into water, the temperature of the water during the gradation test, and the amount of toxic metals before and after using the napkin. Process and development. Mixture preparation. We combined starch, recycled cellulose, cytosine, and algae extract using a mixing bowl. Sheet formation. The mixture was then flattened over a surface using a rolling pin to give the texture and tightness. Drying. Once shaped, the sheets were placed on baking tray and dried in an oven at low temperature until they solidified. Testing and experimentation. The napkin biodegradability was tested by putting small pieces in containers with water and measuring the degradation time. Tools and techniques for decoration. Materials were all added in a mixing bowl. Once the mixture was placed in a blender. We formed a shape of texture and a sheet with a rolling spin. We placed the sheet in a baking tray. For drying, we placed the baking trays containing the sheets in the oven, the thermometer, scissors, and water containers for testing. Improvement statements made. The formula was adjusted by adding more starch to improve the flexibility and resistance. This way, the functionality of the prototype was improved. Variables and measurements plan. What will be measured? The degradation time, the pH variation, the metal absorption capacity, the flexibility and resistance. How it will be measured? The degradation time will be measured as stopwatch and visual observation until complete the dissolution. The pH variation will be tested with pH indication strips. The metal absorption using a colorimetric test, and the flexibility and resistance manual bending and stretching. The purpose of each measure is to confirm the napkin fully degrees in water, to demonstrate that it doesn't alter water composition, to validate the bowl as eco-friendly material, capability of absorbing contaminants, to ensure it remains usable as a real napkin. The prototype served as a physical base of an experimental stages. The control and modification of its composition allows to understand how it affects all the variabilities of measure. The analysis and discussion. The napkin degraded faster in water with higher temperatures, confirming the biodegradability principle. Nature polymerase in the field effectively absorb the heavy metals due to their challenging properties. The results align with literature and absorbing the adaptation currents depending on the chemical composition, pH, and exposure time. In conclusion, we can conclude that the objective of this project was achieved by the creation of napkin prototype capability of both absorbing contaminants present in fish organisms and degrading in water. The result of our experiment aligned with the following theoretical concept: biodegradability, absorption, reaction, and pH activate release. With the results, we can say that the real-life application of the prototype will face one of the most critical environmental changes and society faces: water contamination and the engagement of its species.